Hello everyone, welcome to my simple zoology class. In today's video, I'll be dealing with the topic structure of sarcomere as well as structure of the contractile protein. Okay, so first of all, you need to understand what exactly is a sarcomere. Sarcomere is nothing but it's a structural and functional unit of our muscle fiber. So hence you call it as what? Sarcomere. Okay, here you can see there is a diagrammatic representation of one of the sarcomere. Now, we already know that there are three different categories of muscle fibers, correct? Based on their functions or their origin or their location, there are three different categories of it. One is skeletal muscle fiber, second one is smooth muscle fiber, third one is the cardiac muscle fiber. The diagrammatic representation which is seen on your screen is right now about the skeletal muscle fiber. So first I'll be explaining about the fine structure of this muscle fiber and then we'll be proceeding with the contractile protein. Now, as I already said, we have taken example of which one? The skeletal muscle fiber. Now, questions related to this appears regularly, like in the form of statements, or it can be a direct question also. What shape is it, and is it whether branched or unbranched or striated or non-striated? That means these respective bands, which I'll be explaining right now. Based on that, the questions will definitely come. The first point you have to remember is this particular skeletal muscle fiber is either cylindrical in shape or it can be either tubular in shape. And what type is it? It is unbranched. So please do remember questions on that has been also asked whether it is branched or unbranched, and the answer will be unbranched. Okay. Now the outer membrane of this muscle fiber, that is the sarcomere, is made up of plasma membrane. Regularly we call a cell membrane, right? That is plasma membrane. The same thing over here, you call it as what? Sarcolemma. Plasma lemma or plasma membrane. Anything is fine. Similarly, you here call it as what? Sarcolemma. Okay, and the membrane of the cell of this muscle fiber is basically made up of what type of fibers? And the answer will be collagen fibers. So please do remember this is very important, that is collagen fibers. Okay, then they also have cytoplasm. Here you don't use the word cytoplasm, rather you use the word sarcoplasm. So you use the initial prefix region as sarco, whether it is sarcoplasmic reticulum or sarcoplasm, sarcolemma. So anything like that, you have to use the word sarco because we are talking about the muscle fiber over here. Okay. Now coming to these lines over here. Okay. Now, what exactly are these lines? These are the protein lines or the protein content of the muscle fibers, and that you call it as what? Myofibril, or the fibers over here, they are called as what? Myofibril. Okay. Now these myofibril, they are arranged in these parallel rows. Okay. So the myofibril over here, they are arranged in the parallel rows in the form of dark and light band. Okay. Now these dark and light band, they are present in the alternate order. Okay. And these dark and light band, they are made up of two categories of protein. One is actin protein, the other one is myosin protein. Which is the first one? That is actin protein, and the other one is the myosin protein. Okay. Now we'll understand what are these actually proteins. What type of filament are they made up of? Okay. They are ultimately filamentous protein itself. Which one? The actin and myosin, which I already said dark and light bands are there. We will distinguish which one is the dark band, which one is the light band, and both of them are present in the filament form itself. Okay, that you have to remember. Now, which are the filaments which are thin? Actin filaments, if you can see over here, the blue one, actin filaments are thin filaments, whereas myosin filament are thick filament. In the diagram itself, they have mentioned very neatly that is actin is a thin filament, myosin is the thick filament. Okay, then light band is made up of only, now I was just saying, right, dark and light bands. Now we will understand which one is a dark band and which one is the light band. Okay, light band is made up of only actin filament. Okay, light band is made up of which filament? That is the active filament, whereas these bands They are also called as what type of band? They are called as I band. I band stands for isotropic band. So please remember, questions on this particular diagrammatic representation will be always asked. Okay, what is I band? What is A band? All these things has to be remembered. Okay, so I'll repeat once again. Light band is made up of what filament? It is only the thin filament, that is the actin filament, and where only actin filaments are present, what type of band it is called as? It is called as I band or isotropic band. Okay, then in between this I band, okay, there is a line passing through. If you can see this dark black one, that is called as Z line. Okay, Z line is going to bisect this thin filament. Okay, so basically this thin filaments, they are attached to this Z line itself, which is going to separate our actin filament on one side, actin filament on the other side. Okay, then Z disc has to be remembered because questions on this has always been asked, and there is another name also, that is called as crosser's disc. So just to remember, what is the Z disc also called as? Crosser's disc. So please do remember that. Then coming to another one, there is another type of band that is called as A band. In A band only comes in the middle, that is where the thick filament, that is our myosin filaments, are held. Okay, I'll repeat the whole thing once again. I band comprises of only thin filament, that is actin filament. A band comprises of what? Only the thick filament, that is the myosin filament, and half of the thin filament. Then there is another disc that is bisecting our thin filament, that is called as what? Z line or Z disc. Anything is fine. Then apart from that, there is another one also here in the center. It is not mentioned over here. If you can see, I'm highlighting, that is called as M line, which is going to bisect our membrane, the thick myosin filament membrane. Okay, so along with that, there is another zone also. The central part of the myosin filament over here, only the myosin filament is called as what? Head zone. If I have to write down everything at once, I band is nothing but what? I band only thin filament. Okay. A band is forming what? Thick plus thin. That is myosin plus actin. Then H zone only thick. Okay, so these three you have to remember, particularly questions on these have definitely come and will come also. Now on a whole, for a sarcomere to be formed, okay, a sarcomere is considered as a functional unit of contraction, right? So this sarcomere has got one A band and two half I band. What does it form? One A band and two half I band. Okay, then one is A band over here and two, this is the one, this is the one, this is the one, this is the one, I band to be present. So this on a whole forms the sarcomere structure. I hope I've made it clear what is exactly sarcomere, what are the questions related to this is going to come. Okay, here you can also remember it as what? Crosser's disc or crosser's membrane. Anything is fine, so need not have to worry. Then for one, this is another one important in the form of note I'll be writing down over here. So please do remember, that is one myosin filament is surrounded by how many actin filaments? This question has come. That is 6 actin filaments. See, one myosin over here. So 1, 2, 3, 4, 5 and 6. So totally how many are getting? Totally 6 of it are going to be surrounding. Similarly, one actin filament is going to surround how many myosin? 3 myosin. The questions on this has definitely come. Please do remember. One myosin filament is going to surround 1, 2, 3, 4 and 5, 6. So totally 1, 2, 3 and 4, 5, 6. Totally 3 of the myosin filaments. 1, 2, 3, 4, 5, 6 of the actin filament. In that way they are considering. Okay. Then one actin filament is having how many myosin? 3 myosin. They are taking on a whole representation. Need not have to confuse. Here 1, 2, 3 myosin. 3 myosin equals to 6 actin. Okay. 1 actin equals to 3 myosin. That way you remember. Okay, then coming to the next one is the structure of contractile protein. Now we already know that there are thin and thick filament. Thin filament is our actin filament, thick filament is our myosin filament, correct? Now this actin filament or thin filament, I'll be explaining both of these in detail. Okay, each of the thin filament is made up of two F categories of filaments. If you can see the diagram over here also is mentioned, that is filamentous form of actins will be present. They are wound to each other, and each of this F filament or the filamentous actin filament is a polymer of which one? Globular. Basically they are in the globule form. Later they change themselves into what? A filamentous form. With the help of which particular ion? And the answer is it's with the help of the Mg2 plus magnesium ion. Question on this also has been come. Now two filaments of these have got another protein also which they are running close. If you can see the red one, that is called as tropomyosin. This is the another protein which is running close to this F actin throughout its entire length. Then coming to the third one, there is a complex protein. If you can see the green ones, they are trimeric in nature. That means three units are combined over there, and the name of that complex protein is troponin. Now why is it called as trimeric protein? Because it has got three subunits present in them, that is TPI, TPT, TPC. TPI is nothing but what? It is an inhibitory site. It is going to inhibit the binding of actin and myosin filament. TPT is nothing but what? TP first of all stands for troponin. TC I'll be explaining. I hope you understood. T is nothing but what? Troponin is going to combine with this tropomyosin. Then TPC, troponin has got calcium ion binding to it. Now why is this calcium binding site present? Because of the presence of the calcium binding site itself, you can expect for the muscle contraction to take place. So when I'll be explaining about the mechanism of muscle contraction, over there you'll understand how this trimeric protein is going to come into action. I'll repeat the whole thing once again. There is G, that means globular actin form is a monomeric form which is going to polymerize to form filamentous category of actin, that is F actin, with the help of magnesium ion. And there is also another protein called as tropomyosin which is going to run along its entire length of this F actin. And there is another complex protein that is called as troponin, which is also called as trimeric protein because it has got three subunits, that is TPI, TPT and TPC. Okay, this is about the actin filament. Similarly, there is another one, that is myosin filament. This is the last one. Myosin filament is also called as what? Thick filament, correct? Now, this myosin filament, they have got many proteins, or you can also say single unit of proteins, monomeric proteins. Now these monomeric protein itself, they are together combining to form and give rise to a structure, that is the thick filament. And because it is going to form one complete thick filament, they have a name that is called as meromyosins. Okay, now each of these meromyosins has got two categories. Okay, you categorize it as HMM and the other one as LMM, that is heavy meromyosin, and the other one is the light meromyosin. Heavy and light. I'll tell you what are these heavy parts, or which are the parts which comes under heavy and which are the parts which comes under light. Okay, now each of this meromyosin, you ultimately call this whole structure as meromyosin itself. Okay, each of the meromyosin has got two important parts: a head region which is globule in shape, or you can rather call it as globular head, and short arm. This is the short arm, and finally this is the tail. Okay, now the heavy meromyosin is the region where the globular head and short arm is present. That is this whole part. If you can see, I put a flower bracket, so no confusion. Whereas the only tail region forms the light meromyosin region. Okay. Now the heavy meromyosin is nothing but what? Head and short arm. So that region is going to project outwards, and here itself you completely get to see the cross arm because of this particular arm which is going to twist itself, and that is going to form a cross arm. Then another important question: what does the globular head of the meromyosin has? It has got two binding sites. One is actin binding site, the other one is the ATP binding site. Okay, during the muscle contraction, actin has to come and bind onto this myosin head. So this is our myosin head, right? So actin has to come and bind onto the myosin head. So there should be some site wherein the myosin is going to come and bind onto the actin. And that site over here is present on the myosin head. That is on the globular head region. So there the actin is going to come and bind, and then you can see for the muscle contraction to take place. Similarly, there is another binding site also called as ATP binding site. Now, what is the function of ATP? Adenosine triphosphate is doing the work of energy releasing. Because of the usage of ATP, which is going to combine or come into action to phosphorylate to form what? ADP and inorganic phosphate. So then you can see for the actin and myosin binding to take place. ATP is used as an energy resource for the actin and myosin to combine and undergo a crisscross formation, and finally our muscle will start contracting. So here how many binding sites are there? Actin and ATP binding sites. So this is about the structure of sarcomere and contractile protein. I hope you have understood. Each and every point mentioned in this note has definitely come in the form of questions. So you make a note of everything. I hope you have understood everything. If you have liked my video, please like, share and subscribe. Thank you.